Welcome everyone, and thank you for being here today. My name is Rihanna Putnam, and I'm the Community Engagement Specialist for the Citizen Science Association. I have a few slides about the CSA and some housekeeping notes, then I'll turn it over to our moderator, Leah Shanley, to introduce our panelists. The Citizen Science Association is a member-driven organization that connects people from a wide range of experiences around a shared purpose of advancing knowledge through research and monitoring done by, for, and with members of the public. Our efforts are concentrated into a biennial conference. Our next one's in May of 2021, a peer-reviewed journal, and member services such as our working groups, online networking, and webinars like these. Our membership represents the multidisciplinary and multidimensional nature of the field and includes project leaders, researchers, educators, and more. Connections are what makes membership to the CSA really important, and now is a great time to join. You can find out more about that on our website. We have a whole suite of COVID-19 resources that we recently published, including some more information about some of the groups that I'll be talking today. You can find that on our website, and we do have a few more upcoming webinars that are part of a series in response to COVID-19 that we'll speak a little bit more about in a minute. This webinar was coordinated by our Law and Policy Working Group. You can find out more about upcoming meetings, past events, and their Ask a Legal Question tool on our website. I'll share the link in the chat. Our working groups are one of the key ways we work to advance the goals of the CSA through the sharing of resources, best practices, and relevant information, both among group members, but also to the broader citizen science community. There are 9 working groups that explore cross-cutting issues like ethics, data metadata, evaluation, and more. Getting involved with the CSA is a great way to make connections, grow as a leader in the field, and help support the association. This webinar will be recorded and uploaded to a YouTube playlist where we archive past webinars on topics such as research ethics, privacy, consent, and more. I'll share the recording via email along with any links shared by presenters sometime next week. We are doing several webinars geared towards citizen science practitioners this month to hone their skills and make connections. You can find out all about our webinars on our website. We actually have 3 next week. We have a data quality Ask Me Anything on Tuesday that might be of interest to some of you, in addition to a book discussion and also a community check-in on Monday. That's a great opportunity to chat with others in the field about your experience during this challenging time. The Law and Policy Working Group has one final webinar coming up at the end of May as a part of their ongoing series. This one will be on citizen science, community engagement, and the UN Sustainable Development Goals. And so that one's on a Thursday at 11 a.m. at the end of May. Though this webinar was free to register, we encourage you to make a donation of any amount if you're in a position to pitch in. We believe that community-supported webinars, particularly those from our working groups and led by volunteers, shouldn't be paywalled. So if you find value from this learning opportunity and have the means, you can help cover the hard cost to keep webinars free for those who can't pay. We are doing more webinars than ever this month and really appreciate your support. Lastly, we do have everyone in listen-only mode today. We do want to get to know you, so if you could head over to the chat now and tell us who you are and where you're from, and we can all kind of find out more about who's on the line today. You'll want to make sure that the chat is set to all panelists and attendees, so that way everyone can see your responses. The chat is also where we'll be addressing questions, and so if you have questions throughout people's presentations, feel free to put them in there, and our moderator will direct them back to panelists at the end. And with that, I'll turn it over to Dr. Leah Shanley, a senior fellow at the Nelson Institute at the University of Wisconsin-Madison and co-chair of our Law and Policy Working Group. Thank you.
Thank you, Rihanna, and thank you to our panelists who are joining us today, and thank you to the audience for participating. We'd like to thank the Citizen Science Association, and particularly Rihanna for her hard work in helping us to make this happen. As Rihanna mentioned, I'm co-chair of the Law and Policy Group for the Citizen Science Association. We've been doing a series of webinars every month since the fall, looking at different aspects of both the legal, policy, and regulatory issues of citizen science. Things like privacy, consent, the policy impacts of citizen science. But with COVID hitting everyone, and this being an enormous both national, state, and regional challenge that policymakers are trying to wade through, they're trying to understand what the models mean, what are the economic impacts, etc. That we thought we would also focus on the citizen science projects related to COVID. So we'll talk about the science today. We'll talk about the challenges of running such projects, and maybe the presenters might also comment briefly about any policy challenges or impacts that they've had. With that, I'd like to welcome the first presenter team for today. Mentioned Dr. Raju Das. He's a computational biochemist at Stanford University School of Medicine. His lab seeks a predictive understanding of how RNA molecules code for complex biological machines. Their lab and his team are developing high throughput, multidimensional chemical mapping experiments to uncover 3D structures and conformational changes of non-coding RNAs in their biological milieu. You guys are gonna have to explain that to me. Dr. Das and our speaker today, Do-Soon Kim, who's a PhD grad student in the lab, are leading an Eterna massive open laboratory that couples a 250,000 person player video game to the lab's massively parallel experimental tools and deep learning, the first such platform in citizen science. So I am so excited to hear this presentation. And with that, we'll hand it over to Do-Soon Kim.
Hi, everyone. I will share my screen and I'll jump right to it. Cool. Hi. So thanks for the introduction, Leah. I'm thrilled to be here to talk to you today about Eterna, and specifically our effort to develop a COVID-19 vaccine by sort of crowdsourcing scientific ideas, and also including, uniquely to Eterna, including experiments, wet lab experiments directly in the loop. So I'm thrilled to be here and talking to you. I'm happy to field any questions, and I'll be around to answer questions after. So this is an iconic photo. This was taken 63 days after the release of the genomic sequence of the coronavirus, the virus responsible for COVID-19. And this is a picture of Jennifer Haller, mother of two and a nurse from Washington, receiving the first human dose of a new technology called mRNA vaccines that can hopefully build immunity to the coronavirus. And what's remarkable is that it went from 63 days from the genomic sequence identification to clinical trial ready vaccine dose, which is a remarkable time frame for vaccine development. It's almost unheard of time frame for vaccine development. So we at Eterna looked at this key new technology, mRNA vaccines, and knew this might be exactly in our wheelhouse to help in this effort of designing this mRNA vaccine. So we launched this new campaign called the OpenVaccine campaign. And our mantra is a COVID-19 vaccine for everyone, designed by everyone. And I'll tell you a little bit more about the Eterna platform to sort of explain what we mean by this. So on Eterna, the overall platform goes by this other mantra, played by humans and scored by nature. And what we mean is video game players using a web browser and by logging into the Eterna game platform can design RNA molecules in real time. And they can design tens to hundreds of these. And then the same players will go and they'll vote for their designs. They can go through our data browser, see what they've designed, see what other players have designed, and they can vote on the designs that they like the best. We then, and this is a really key part of Eterna, we then take those RNA molecules and we actually synthesize them and do biochemistry experiments, some of which Leah might have mentioned in the bio for the DOS lab. They actually do those synthesis experiments to characterize the RNA molecules that the players have designed. And then we return the results back to the players who are designing those molecules in the first place, so they can iteratively learn in cycles from the results of their initial designs. So what we've done is, in short, crowdsource the scientific method. But instead of a team of 10, a team of 20, and maybe even 100 in a laboratory somewhere, it's actually a team of 200, tens to hundreds of thousands of players that are sort of learning from their experiments and design principles. Some of the accomplishments that we've achieved through this method are designing thermodynamically perfect riboswitches. So this is something that even the state-of-the-art computational packages can't accurately predict, but Eterna players have been able to design almost perfectly functioning riboswitches. We're also working on a platform for designing RNA in 3D. So in nature, RNA molecules are not so planar like you might see in the current video game. We're working on efforts to bring 3D design to life. We also participated in an effort to make gene editing, this CRISPR technology that's been really fascinating and potentially powerful, to design the guide RNA molecule, key RNA molecule, and the mechanism to be very safe so that gene editing efforts can proceed with more on-off switches, more tighter on-off switches. And then finally, and importantly, Eterna players have become scientific contributors to publications that have been peer-reviewed and published for the rest of the academic community, as well as the broader public, to read and learn about. And now I'm going to dive back into sort of the specific challenges of realizing the OpenVaccine campaign. And this sort of ties back to what Leah said about the policy portions and the more platform-wide concerns that we may have about the citizen science platform, and specifically our video game citizen science laboratory that we run. The first is identifying the right and new puzzles for players. So this is where a little bit of scientific expertise comes into play. And you want to give the players a good target to design towards. Many of our players don't have formal scientific training, and this is a great sort of leeway into gaining a scientific training and familiarity with the science that we're doing on Eterna. So designing the correct puzzles for the players to solve and designing the right RNA molecules is really key. The second is expanding playability and accessibility to Eterna. And this is something our software developer team, which is just full of amazing software developers, are working very diligently on, which is, so Eterna is available to anyone with internet access, a computer, and a web browser. But what if you live in a part of the world that doesn't have that? How do we bring Eterna to players whose main method of connecting to the World Wide Web is their phone? So we're developing mobile developments. We're making improvements to our sites in terms of performance to make sure people with less access to technology and the internet all over the world can also be a part of the Eterna platform. The third is making sure those experiments that players design are done well, and to develop new techniques to handle the volume of designs that we may be seeing. So this is done principally by scientists and researchers at Stanford University right now, who are developing new techniques and also going into the lab every day to make sure that from the wet lab side, we can diligently test the designs that players have submitted to the Eterna platform. And finally, and especially with the OpenVaccine campaign, this has become incredibly notable to us how important this may be, are industry agreements. And what we mean by this, Eterna can do the discovery and preclinical side decently well. Like that's what we're really good at, is discovering new molecules and coming up with new and better designs for RNA molecules. But to bring this idea and an academic laboratory finding to the real world, to get a vaccine so that we can start vaccinating people with the proper doses and the proper safety controls in place, we're going to need industry partners and industry agreements to make sure that we can safely bring the vaccine out and scale up production so that a lot of people can have access to the vaccines. So this has been a really key component that we've been working diligently on, is forming the right industry agreements and looking for the right partnerships that can bring Eterna to the real world. Finally, I want to acknowledge everyone who's been involved. This list is not exhaustive. More broadly, the DOS Lab, Barna Lab, and Professor Pasu Huang's lab at Stanford University have contributed greatly to the Eterna platform and also specifically to the OpenVaccine Challenge. I list some experimental scientists as well as computational biochemists here. Individually, Professor Howard Chang's lab, Hani Chowdhury's lab at KAU, and Baidu Research USA. I want to thank them for their contributions and support of the Eterna platform. And of course, the Eterna developers. Danny Braun is here today, as well as some Eterna players, hopefully, and other Eterna developers. It's their work that really allows the Eterna platform to flourish. I want to give a lot of credit there. I've noted just a few Eterna players that have directly contributed to something in today's presentation, but you can see on the bottom right that these are some of the screenshots of all the fun player icons and player mascots that they use on the Eterna platform. And they've been instrumental in pushing the platform forward. And on the top right, we want to acknowledge some financial support and also partnerships that we formed over the years that make the Eterna platform possible.
Excellent.
Lastly,
go ahead.
Sorry, I just want to say lastly, please engage with us. Please go to eternagame.org, sign up for an account. You can engage with us we are expanding our social media outreach, so feel free to engage with us on Twitter or YouTube or Facebook and Instagram. Thanks so much.
Wonderful, fantastic. Really enjoyed that. So we're going to save questions to the end, but people can start to submit their questions via the chat box on the right. And panelists, you can start to answer those questions, but we will also talk about those questions verbally at the end. So with that, I'd like to now introduce Dr. Firas Khatib. I'm sorry, do I have that correctly?
Perfect.
Okay, thanks. He's an assistant professor in Computer and Information Science Department at the University of Massachusetts, Dartmouth. Firas is the lead scientist of Foldit, a video game that has allowed hundreds of thousands of players to contribute to biochemical scientific research. The long-term goal of this project is to utilize the combined power of humans and computers in order to build accurate models of disease-related proteins. So with that, I'll hand it over to Firas.
Thank you so much. And I actually just found out earlier this week that I got tenure, so that's why I had to change it to associate professor. So I'm going to talk today about Foldit, which is a free game where citizen scientists from around the world all collaborate and compete to fold proteins and try to get the best possible score. The higher the Foldit score, the higher the stability of the protein. And Foldit is free. It's run by academic scientists. It's not for profit. You can download the game on our website for free. And there's no experience necessary. We don't expect people to have a biochemistry degree and know anything about proteins. We really just have you focus on just trying to get the best and highest possible score, which has all these very complicated calculations run behind the scenes. Now, that doesn't mean that you just jump in to Foldit and start fighting COVID-19. The first thing you're going to encounter is a bunch of tutorial levels, such as this one in particular for protein design, where you can suddenly design new proteins that don't exist in nature. And you can imagine that this is not an easy thing to do. In fact, in a couple weeks, Foldit is going to be celebrating its 12th anniversary. Yet we struggled with protein design for pretty much most of those 12 years until a breakthrough really last year, where suddenly our Foldit players were able to design brand new proteins that do not exist in nature. And that's their designs here are the ones that are shown in color, where we then solved those corresponding new proteins, and they folded up exactly the same way, which you can see here in the gray. And these are just zoomed versions that show you how accurate the Foldit player predictions were. But again, this didn't happen overnight. In fact, most of the years involved Foldit players discovering many flaws in our energy function and finding essentially many bugs that had gone unnoticed, because humans will try out things that are automated protocols will not. So this was very exciting that they would be able to design new proteins and have them really fold up the way that they have predicted. And so the new challenge is, of course, to design proteins against COVID-19. And so we recently launched 2 different challenges in Foldit, trying to come up with 2 different strategies to combat COVID-19. And so the first effort is to design an antiviral protein to block COVID-19's spike protein. You've probably all seen this artist rendition of COVID-19 with all these spikes around that form a corona, hence the name coronavirus. And so our first goal for Foldit players is to design a new protein to block those spike proteins, which you can see here in the game. So we've taken the spike protein, the actual exact same one from the self structure, and now our Foldit players are trying to design proteins to inhibit or block those spike proteins. If they're able to block it, then the COVID-19 virus wouldn't be able to attack our body. So that's one of the 2 challenges that we're doing. The other challenge is to design an anti-inflammatory. So you might have heard of the term cytokine storm recently, which happens when the immune system releases more and more cytokines to defend itself, shown here as these white dots. But instead, this causes the immune system to go into overdrive, and it starts to attack human organs. So the approach we've taken with Foldit is to try to block these cytokine signals in order to stop that overstimulation of the immune system. And so this is a very similar problem from a Foldit point of view. You know, we just need to design these green dots here in order to block those white cytokines. Now, easier said than done, of course. This is a very challenging problem because designs need to have all these very, very many small interactions with their target, right? And so, you know, those have to fit perfectly. You can almost think of it as keys and locks and which is why we believe that Foldit is such a good puzzle, is because this is really a three-dimensional puzzle that we're trying to solve. And humans generally are very, very good at spatial reasoning and pattern recognition, and that's one advantage that they have over, say, computers. You know, visual tasks is one thing that humans are still better at than computers. This is why we have CAPTCHAs whenever we're on the internet. We have to prove that we're humans. The other challenge is that not only do they have to have very good interactions with the target protein, the Foldit designed protein has to be able to fold up by itself as well. So these are two very difficult elements that need to happen if it's going to be successful. So Foldit players just started tackling this anti-inflammatory strategy, but we have begun testing 99 Foldit player designs from our anti-viral puzzles. This is just a couple weeks ago, and this is starting from over 20,000 unique Foldit submissions. And you know, the one thing I want you to notice, you know, you don't have to stare at every single one of these proteins, but you can see the diversity among them, which is really the exciting thing. You know, these are all very, very different topological approaches to blocking the spike protein on COVID-19, which is really you know, exciting. And what I think about is, especially with citizen science, you know, a way that it's very powerful is it's looking at problems from very different angles. And we've seen this a lot with our Foldit players. You know, one of our most successful cases where they were able to solve a protein that hadn't been solved in over 20 years, and you know, they did it in 10 days, was they completely thought outside of the box and did it in a very different way than the traditional methods. And so that is very much our hope with this project. And one of the great things about Foldit is how much of a collaborative effort it is, not just with our citizen science scientists, of course, but with the amazing Foldit team, which is scattered throughout many universities. So the Institute for Protein Design is where all this lab work is being done. And we have these great videos by Brian Kepnick, who explains these puzzles and what's going in the lab in very, very accessible ways for anybody really to contribute to the problem. And the game is designed at the Center for Game Science at UW, Northeastern, University of Denver, Vanderbilt, and of course, most importantly, without which none of this would be possible, the Foldit players. I can never thank them enough. So please, you can try out the game at fold.it. We have a Discord server. This is our YouTube channel with all these really, really great videos. We have Twitter, Facebook, and all that stuff. But yeah, thank you.
Wonderful. Thank you so much, Firas. Excited to ask more questions. Again, folks can enter their questions for the panelists in the chat box, and then we will address them at the end of the presentations. With that, I'd like to invite Dr. Kara Seawalk, MPH. Kara is a health map project manager for the Computational Health Informatics program at Boston Children's Hospital. She's got a background in infectious disease epidemiology and manages the digital health initiatives for the group with a focus in disease surveillance and public health promotion. During this COVID pandemic, Kara and her colleagues have launched COVID Near You, which is a citizen science website aimed to increase signals of COVID-19 for public health surveillance and research, building on the Flu Near You effort they'd had. And they're using participant-reported symptom and behavior data. With that, I'll hand it over to Kara.
Hi, everyone. Thank you so much for having me, and thank you for that introduction, Leah. I'm really excited to be here to talk to you about our COVID Near You platform and why it was created and what we hope to achieve. So as Leah mentioned, a big part of our research lab at Boston Children's Hospital is designed to support public health initiatives around surveillance of infectious disease. And a good example of that is with our Flu Near You platform. In 2012, our team of researchers launched Flu Near You, which is a participatory influenza surveillance platform to engage the public in public health initiatives, for them to provide symptom-reported data to support public health surveillance of the seasonal flu. And so since 2012, the Flu Near You platform is run based off of reports submitted weekly by our cohort of reporters to provide on-the-ground insights into what is affecting the United States in terms of flu symptoms. So with this type of technology that we've built, we decided in January and February that it would be a really valuable type of tool to pivot for the COVID pandemic. So a little more background on Flu Near You. This is one example of how we compare our Flu Near You ILI, influenza-like illness surveillance data, to traditional public health measures from the CDC across multiple flu seasons, so that we can see how it compares, particularly because the Flu Near You population is unique in that we're capturing information from people who may not be seeking medical attention, and we're also providing this information in real time, so it could give us an idea of what's happening right now in this given week for flu surveillance. We've also published a number of manuscripts on the research from Flu Near You on crowdsourced symptom reporting across influenza seasons, and exploring who this population is and how it can provide value in a citizen science type of initiative for flu surveillance. So again, with this background, we really wanted to pivot that type of research and that type of technology to see if we could provide some insights into the COVID pandemic. And so we felt as a research team that it would be more beneficial to pivot this technology and brand the product for COVID so that we can engage a larger percentage of the population to contribute into COVID surveillance directly and report their symptoms. A big challenge that we've seen, and I think everyone here probably knows, is that not enough Americans are getting tested for COVID, and that's a real challenge from an epidemiological perspective of not knowing the true impact of this disease. And so using, crowdsourced, participatory reported symptoms, we can try and get an idea of what's actually happening among those who are not seeking that medical attention. So kind of a little background on how we got started. Our research lab does run Flu Near You and continues to, but we have partners across the technology sphere who really wanted to contribute to this research initiative and who thought that this type of participatory surveillance could be really beneficial in terms of understanding the true burden of this disease. So in March 2020, we partnered with a number of tech companies across, from Amazon to Apple, Google, Airbnb, et cetera, to really pitch in and volunteer their free time to help us get this platform built up and out the door for us to promote to the public and start utilizing for surveillance measures pretty quickly. We got it up and running in about a week, and then our team internally at Boston Children's continues to maintain the platform. So it was a bit of a whirlwind to get it up and running, but it was really crucial to us to support this initiative and see if we can find any epidemiological information from the data that we're collecting. So before I get into what exactly COVID Near You is, just as a quick background for what the platform is and how it runs, I wanted to highlight why this type of information is needed. As I mentioned, there is a lack of extensive testing happening in the United States, and this type of information can really provide some insightful data where it's currently unknown. And providing this real-time information, again, is going to be capturing people who may not be seeking medical attention at this time. We're also collecting information on quarantine behavior, travel behavior, whether they've gotten the flu vaccine. So really trying to capture as much information from users as we can to hopefully provide some insights for the pandemic. And this type of novel data can help us potentially identify current hotspots and identify potential future hotspots as well as we see the pandemic continue to move forward. So I'll just quickly run through how it works so that you have an idea of how this type of citizen science project runs. It's a simple website. You just go to covidnearyou.org. And we have a very simple question. How are you feeling? To us, in terms of epidemiological research, it's just as important for us to know who's feeling well as who's not feeling well to capture both sets of information. But when you do enter that you're not feeling well, we ask what symptoms you may be experiencing. And that can range from fever to fatigue, cough, nausea, loss of smell or taste, loss of appetite, etc., for the users to provide us some information on what they're experiencing. Again, this is where we ask that additional detailed information. We ask what date did they start feeling ill, because the date of symptom onset doesn't always correspond to the date that they're filling out this application. We have had a number of people reach out who want to provide information that they're experiencing or have experienced 3 weeks ago, 4 weeks ago. So indicating what that first date of symptom onset is critical to understand how long this has been happening for. And then we do ask what type of health professional they saw, if they saw one, and that can range from a doctor, a virtual visit, an emergency room visit if they've been hospitalized overnight, etc., so that we can kind of get some information and insights into healthcare utilization among this population as well. We ask information around if they've been tested for COVID and what those results are. And from our research perspective, it's really interesting to see if you are confirmed as a COVID positive person, what are the symptoms that you're reporting in? We all know that the kind of strict guidelines around how you define COVID-like illness is limited to fever and cough, and fever and shortness of breath. But there are many symptoms that have been coming out from physicians across the US and across the world that don't correspond to those strict three. And so it can give us some type of insight into what other symptoms are we seeing emerge among the United States population. We also ask, again, how many days we've been in quarantine, if you've traveled outside the United States, if you have, what was the last country you traveled to, and if you've had known contact with someone who has been a confirmed case of COVID-19. We also ask for demographics, so age, gender, zip code, all of which are incredibly useful to understand the populations that this disease are impacting, particularly those who are reporting into our site. And this type of information is really interesting to compare geospatially. So how does this differ across the regions that we're examining? And then last but not least, we ask the users to submit their phone numbers so that we can continually remind them to report in. This type of surveillance is incredibly useful when we have longitudinal data of reporters coming back every week or every few days to report in how they're feeling, so that we have longitudinal information captured from each user. So, and this also, this type of initiative of submitting your phone number is really great for citizen science projects because it really does engage the users to continue to come back and feel like they're contributing. And we find a lot of value in having that reminder to the users to continue to report in. So, you know, capturing the information is great, and it definitely supports our research initiatives around you know, what we're hoping to find. And, provide to public health officials. But just as important to us from a citizen science perspective is also showcasing the results to our users so that they can see where their data is going, and so that they can also have a tool to interpret the data as well, so that they can see, the information they're reporting isn't going into the cybersphere and kind of lost. We really wanted to provide that information back to them. So we provided this mapping of symptom reports. And right now our team is actually working to optimize this mapping experience to make it a little bit easier to use. But for the first iteration, when we first launched this project, we wanted to map those who are reporting into our site, who are reporting the COVID-19 symptoms, which are the fever or cough, fever or shortness of breath. One of the additions we're making is to expand that symptomatology of mapping for other symptoms that could be related to COVID as well. And then we're also mapping those who have tested or reported being tested, so we can get an idea of where tests are being taken. This was really interesting to map at the beginning of when we first launched this project in about a month ago in March, because as we see now, tests are becoming increasingly more available, but a month ago was extremely limiting. So getting an idea of geospatially of where tests are being administered to this population was really important to us. And then we also did want to provide information to the users on COVID activity in the United States. So then comparing that to confirmed cases across the US and confirmed deaths as well, so they can have more of that confirmed information from validated sources like CDC. And then we also allow users to report by zip code. So here you can see, if you type in your zip code, you can get a sense of the activity that's reported on this website in your area. And this, I think, is even more important for the user because it can directly affect them, and they can get some insights into what activity is being reported in their neighborhood. So having that kind of more granular detail was really important to us. So we've had over 750,000 reports collected. This spans across our platform. So we first launched in the United States, but we've emerged from across North America with interest to expand the platform. So about a week and a half later, we launched the platform for Mexico and Canada as well, so that we could support surveillance measures for both Mexico and Canada in conjunction with the United States to see how it impacts North America altogether. And so we're some really interesting data come out from all three countries, and it'll be interesting moving forward to compare and contrast as the different nations have different response measures to the pandemic as well. So from our epidemiological research, we've started to dig into the data a little bit to see what our initial findings are. And this is just one example of how we're mapping the data geospatially. We're taking that CLI variable to see if we could geospatially differentiate across the United States. And this will continue to develop as we move forward into the pandemic as well. And we've also created some research around understanding how medical visits vary by symptoms reported and by age group as well. And so we can see here for medical visits, those who are reporting both symptoms of cough, shortness of breath are seeking medical attention more than those who are just reporting, for example, and that will change across the type of medical care visited as well, from any medical visit to urgent visit to emergency visit. And then we've also, on the right here, you can see a map of how we've kind of geospatially differentiated between reports per 100,000. And you can see areas where we have significant reporting. There's pockets of the Northeast. We're based in Boston, and California and Wisconsin have a lot. Whereas maybe in the Southeast, we're not seeing as many reporting coming in. And from a programmatic standpoint, this type of visualization is really, really important for us to see so that we can target areas where we need to increase participation and where we can try and spread the word about this platform. So last but not least, I wanted to talk a little bit about our team. So this is the team who put the project together. These are all staff members of mine at Boston Children's Hospital. And what's really important that I wanted to highlight here is, our team isn't just researchers. It's a team of project managers, epidemiologists, software developers, data scientists. So it's a really eclectic group of people with a diverse background of expertise that came together to build and maintain this project moving forward. So with that, I think I'll close, but I'm really happy to share this platform with you and to answer any questions that you may have. Our website, as I mentioned, is covidnearyou.org, and you can go there today to explore. Or you could reach out to us on our email or on our Twitter account as well.
Great. Thank you, Kara. We greatly appreciate that. I've been scribbling down a few questions I have myself, so looking forward to that. If folks, again, could enter their questions into the side chat box. I see one already for Kara's talk. So let's keep moving. So we've got Dr. Črtomir Podlipnik is an assistant professor and faculty of chemistry and chemical technology at the University of Ljubljana, Slovenia. He conducts research of relevant biomolecular systems, including Ebola and COVID-19, together with colleagues and with support to Central Technical Library at the University of Ljubljana. He organized the citizen science project COVID.SI. So with that, I'll hand it over to him.
Thank you for a nice introduction. So I'm from Slovenia, and Slovenia is only country that have law in its name. So, and we will start with old wisdom of Sun Tzu. He told that if you know the enemy and you know yourself, you need not fear the results of 100 battles. So we organized the citizen project from scratch, and with catalytic support of Central Technical Library. So as I said before, that if you know the enemy, we start to study the corona stuff in the December, and we follow literature, media reports, etc. And in January, we decide to create some joint web-based workplace. In February, we went more broad, and we set up web presence and set up social network. And now our work is underway. We had some problems, but we solved this. And now we are expecting first results of our project. So in first stage, we identified the problem. So here we can see the virus life cycle, and we need to answer to some questions: how to disable coronavirus in the most effective way. So what are possible targets for drug design, and which methods are useful for running the drug design project. So we decide to use virtual screening. So here we have a box with keys and a lock, and the structure-based virtual screening is like looking for a key that opens this lock in an Olympic swimming pool filled with keys of all shapes and all sizes. but there is no guarantee that right key is in the pool. So here we can see the concept, very old concept of virtual screening. So we are searching for the ligands that are complementary to the protein target. The important things that we do is to set up community. Okay, normally communities like this, but nowadays communities like this, it's more virtual community, but with real persons behind. So in our community, we have some scientists, so some IT specialists, gamers, biomedicinal specialists. We have also old wisdom. We have students. We have also businessmen and also some housekeepers. So our community is very broad and we share experience with each other in one goal to fight against coronavirus. So one of the main action of our work is distribute computing. So we here is our scheme of community computing. So we have data server where the targets and ligands are located, and then all users are clients. So we have very different platforms, and the server sends data to the client. On client, some calculations are performed, and from client to data server, there is a flow of poses and docking scores. So another wisdom from Sun Tzu is that strategy without tactics is the slowest route to victory. Tactics without strategy is the noise before defeat. So we try to fight to coronavirus in different way. First strategy is prevent virus entry into the host cell. Second is prevent virus to escape from endosome. Third one is disarmament of viral proteases. And here we have some recent results. So we search currently in our data set of compounds, we have 10 million of compounds, but we are working to a really huge database that will contain about 1 billion of compounds. So these results, we got some ideas, and some of these results will go to the tests very soon. So this is this, okay. So this is the end of my talk.
Wonderful. Well, thank you so much. So if anyone has questions, enter it into the box. We've got one more speaker, and then we'll move towards discussion. With that, I'd like to welcome our last speaker, Dr. Angela Eaton. She leads Safecast's air quality and radiation monitoring efforts here in the Americas, supporting participatory environmental monitoring throughout the Americas. Currently, she's working on COVID-19 response through the creation of a crowdsourcing testing map and a virus progression diary. And with that, I'll hand it over to Angela.
Thank you so much. I really appreciate the doctor, but I'm not a doctor. So this is a BGIGI, and it was designed after the 2011 Daiichi Fukushima meltdown in Japan after the tsunami and the horrible experience with the nuclear power plant. So why am I showing you this in relation to COVID-19? It's because this came together after several months of work for Safecast volunteers and people who were energized around finding what they could for their environment. Safecast is an environmental monitoring platform. And right now we are responding to COVID-19 as well as people who want to monitor their environment for radiation and air quality. And so the reason that I'm showing you this is that similarities were existing between the events of 2011 and what we're seeing now in 2020. A lot of fear around the unknown for both radiation, what was happening back then, and right now with the invisible threats of the virus. Back then in 2011, the Japanese government didn't provide any timely or critical information. And right now we're also seeing some missteps and faltering about how to release information to the public. So if the government doesn't have accurate information and media doesn't have accurate information, everybody's searching, who does? One of the ways that we address this is by looking at how people can contribute to the information that's out there and log their own experiences. So with this one little machine, we built a web of trust. And this is a visualization of people doing independent monitoring all over Europe. And you can see the differences in background radiation in parts of Germany and in France. These are submissions that are made to the Safecast platform, and about maybe 200,000 measurements per month are submitted. So I just want to remark here that this is not Safecast data. The point of Safecast, one of the operative foundational beliefs of Safecast, is that all data would be in the public domain. So this is really community source data that is being hosted or published on the Safecast platform, and that's a really important part of how we view ourselves and how we view information. So here we are right now in 2020, and the questions that we're asking ourselves around COVID-19 are: what information exists and who holds it? What information does not exist, and what can you personally do with that information? So this is a COVID-19 testing map that we recently published on the Ushahidi platform. So Ushahidi is also an open source project that is based in Kenya. What we're trying to do here is prioritize transparency and to help people to describe their own experiences with either seeking testing or getting testing. We want people to be safe. We want to really highlight with this that it's important to build trust. And the way that you build trust is that you are able to tell your own story, hear other people's stories, and then validate that against what's being seen publicly, either with the government or through media. So that's a lot of really what's important about communicating out your personal story. Here you have somebody who's submitted their personal experience in getting tested with their grandson. So, you know, once again, one of our founding beliefs is that people should have access to reliable and accurate information in order to make decisions about their own safety, of their friends and themselves and their families. So the testing map really allows people to anonymously post their experiences in attempting to get those tests and interacting with the medical establishment. So here are, this is what happens when tests aren't available or when testing is ambiguous, incomplete, or there's bad information. If you're dependent on single sources of information, of official information, then it's really hard to establish that trust that we're talking about. Here you see people in Spain trying to find tests. This was during the height of the experience, the pandemic in Spain. In Germany, a lot of confusion about how they could not, like why they were refused testing if they had symptoms. And in the United States, when told that you could actually access tests, this is not too long ago, that it still was very difficult to actually get those tests. And also note that these are anonymously published, so that way people can get out what they need to tell of their story without necessarily creating any kind of public interactions that they don't want to have. So open information also helps to reduce anxiety and build community trust. Right here, we're seeing successfully tested people logging their experiences in Australia and in Switzerland. So what I'd like to point out here is that, you know, if you're reading some of these captions, that people aren't necessarily stopping with, yes, I got tested, or no, I didn't get tested. They're giving a lot of information. I think that relieves people, and when other people see those stories and share those stories, that allows them to also communicate and share experiences in a way that provides a general relief and creates kind of a community bond. We noticed that too, and we wanted to point out that testing is not the end point. Testing is incredibly important, and we understand it's important in containing and maintaining the health of a population. But what we're trying to do here is to get feedback on what is useful to people, and once again answer those questions on what information exists, who holds it, and what can you personally do with that information. So as an environmental monitoring platform, we are trying to log the experience of the trajectory of people experiencing their illness. This is another response that we're having to, you know, how to help people with their experience in COVID-19 and how data might contribute to that. Once again, we're an open platform, and we're designing around the needs that we're seeing emerge, rather than trying to design around needs that we're guessing at. Here, we're just listing what our current needs are. We invite you to join us as participants, as Safecasters, and hopefully we'll see you on the testing map, or we'll see you soon. Thank you.
That's fantastic. Thank you, Angela. We greatly appreciate it. And thank you all to our wonderful speakers today. With that, we're going to open things up to questions. So I've already seen a couple come in. And before we do, those of you who have been panelists, please provide links to your projects, to published peer review papers that you may have that you wanted to call out. I know a couple of you called those out in your presentations. And if you're comfortable with it, to your email address, so people can get in contact with you directly if they want to contribute or collaborate in some way. We had one person write in, if I can scroll back and find it a little bit earlier, during Kara's presentation on COVID near you is they mentioned that there's a UCLA project that is doing something similar. And so the question is, we've got several projects perhaps that are all collecting this information. Are there ways for collaboration and synergies across these projects? Perhaps, Kara, you want to comment? And maybe also Angela, because it seemed to align, and Črtomir seemed to align with some of your projects. So Kara first.
Yeah, I am a little bit lost. So.
Okay. Well, then why don't we let Kara jump in? Kara, you've got to unmute yourself.
Aha, I need to unmute myself.
Okay.
So I'm here, yeah.
Okay, Črtomir, let's let Kara Seawalk jump in. She was about to respond.
Sure. Sorry, I was on mute. My apologies. Absolutely. I mean, there's definitely a number of tools across the United States that we've seen pop up, and there's definitely interest globally for this type of surveillance. We are definitely open to collaboration, and we're speaking with a number of groups to see how we can share our data, how we can have other groups share their data, because at the end of the day, we do want this to support public health surveillance and also research on COVID directly. And with crowdsourced data for symptom surveillance, the more data you have, the better it is in terms of understanding the true impact. So absolutely, there's a number of groups. We're working with a lot of them. And if you work for one, or if you know of one who want to reach out to us, we're more than willing to collaborate. It's an exciting field.
Angela, do you want to comment as well?
Absolutely. So the operative part of our platform is that it's open, that all data is open and accessible to anyone who wants to use it. We encourage that use. So it's not just, oh, put your story out and then it goes nowhere, or, oh, put your story out and only your friends get to see it. I mean, the idea is that with more people telling their stories about testing, or as we further develop it and respond to how people want to tell their stories or tell their experiences or journal their trajectory of illness, that can be used by researchers in the future, that can be used by public health officials or people looking to maintain or change the trajectory of the illness for all of us. But it's only if you have that data open and available from the get-go and as a basis that you can really share it on that level and not have it siloed. So that's the thing that we're trying to do.
So back sort of in 2010, 2011, 2012, early days of social media curation for disaster response, the Banda earthquake, the Banda Aceh, the earthquake in Haiti, the tsunami in Japan, we had 1,000 flowers would bloom around crowdsourcing missing persons data. And all those projects eventually got together and formed a consortium and developed a set of common standards and protocols so that even though you had multiple sites, all were collecting similarly so you could aggregate the data and information. And maybe that's something that over time could be developed here. Anyone else want to add a comment to that before we move on to the next question?
I actually, one thing that I think is interesting is how this data might interact with other data sets. So for instance, Safecast has the radiation and the air quality data sets. One thing that I'm particularly interested in is how the experience of COVID on a worldwide scale like this is going to affect those other data sets. So how will it experience air quality and air quality health? Both the negative and the positive of that. So we don't know yet, and we're closely looking at how people's responses and how people's postings can actually intersect.
Excellent. I see the question around volunteers and volunteer management has come up a couple of times. And this is something I think all of you could comment on, but we'll go in order. One of the two or three people have asked about, are the scientists volunteers? You know, who's volunteering? How does that management of those volunteers work? And you guys, I think for us, you guys have been around for 12 years. So how do you sustain that interest over the long term, not just in the heat of a crisis? So we'll start with Firas and then go down to all of you.
Yeah, that's a very good question. We actually were very fortunate in the early days of Foldit in that we had a distributed computing project that still runs called Rosetta at Home, which was just a screensaver. You just donate your CPU time to help fold proteins, and so from there Foldit started, and so we had this very, very large influx of players at that point, and the majority of all the Rosetta at Home, and there's millions of Rosetta at Home users, the majority of them tried Foldit and said, "Whoa, this is really hard. I'm going to go back to just donating my computer time and running the distributed computing project." But the ones that stayed actually are still playing, which is really what we've learned. Yes, it would be amazing if we had millions of Foldit players that contributed, but for these problems, it is quality, not quantity, that matters. That being said, we still, on the contrary, are always trying to welcome new players because, let's say 1,000 new players show up one day, maybe just one or two of them will turn out to be able to become, someone who can contribute to the project. But yeah, it is tough. Basically, the way we've had it is anytime we had some sort of little kind of publication or breakthrough or something, we get an influx of new players and we try to hold on to them as much as we can. And that's also has a lot to do with how we design the game. If those tutorials that I mentioned are not designed properly, you're going to lose so many people, at level 1. If you can, instead of having it a very steep learning curve, make it a little bit more gradual.
Flattening the curve.
Right. Well, and I think the most important thing is actually the community aspect of Foldit. I think if it was just a game on its own where you just did what you could, it would not be successful, because the fact that there's a chat, the fact that you can collaborate and form teams with other Foldit players that then compete against other Foldit teams. The human competitive element is very important, but that camaraderie and that's a very important aspect. I actually drove once all the way to Connecticut because there was a Foldit team meeting, and they invited me, and I got to meet all the players, and it was as if they were old college friends, even though they only knew each other from this one game. So that's a very important aspect to try to somehow instill into the game. And I think Eterna is the same thing, right, Dusun?
Yeah, we face. Yeah, we face a lot of the same challenges that Frost talked about. We get these surges of new players when these acute events happen, and I think the key for us is to keep the players engaged as much as possible, even if there wasn't a global pandemic around. And that happens through scientific collaboration. So the more RNA expert scientists we can bring onto the platform, the more interesting problems they can identify for us to solve. And then similarly, like Frost said, having a really good tutorial and structuring a community such that players aren't thrust into this world of go from a video game player who's curious about bioscience all the way to, hey, design this therapeutic. That's a huge leap. So it's a mixture of, turning scientists into gamers so that the expert scientists can sort of speak gamer and know how to interact with that community. And conversely, turning some of those gamers who are very interested in the science into scientists. And that sort of two-way junction really allows for a vibrant community. And there's a lot of community upkeep that we try to do with players on Eterna as well.
We've had players basically tell us, the game has made me want to go back to school, and going to pursue degrees after playing Foldit for a few years, and that's basically as good as it gets.
Yeah, that's fantastic. Do you bring them in once they've finished their degrees and they become advisors?
Well, definitely. I mean, they are experts in their. Actually, the proudest moment, I think, at least for me, was with that Nature paper last year. That was the first time that we had Foldit players as co-authors with their actual names on a Nature publication. Previously, we'd have them as, you know, Foldit players as the co-author list, but this was, you know, the actual names of the players that designed those new proteins that, you know, so that was really cool. I guess the next step is just for one of the players to win a Nobel Prize, and then we'll be good.
I noticed that Dusun had mentioned something here about the end user license agreement and data that all players must sign. Can you comment on that briefly? And then if anyone else in the panel also has data agreements or things that you want to mention in terms of a policy standpoint.
Yeah, I think specifically to Eterna, and this is a key aspect of citizen science, is that you sort of buy into this agreement that this is going to be a collective effort, that my Zoom is, okay. So you sort of, we have to get the players and the volunteers who are participating in the platform to buy into this collective effort by everyone and agree to be collaborative. And that means sharing data openly, at least within the platform. And I've noticed, you know, Kara and Angela, you know, their platforms are essentially contingent upon open sharing of data and proper anonymizing as you would like to, you know, make sure you don't violate HIPAA, but also on Foldit and also Eterna, you can imagine if players make a discovery that ends up being a therapeutic molecule that has commercial impact, how do you handle that? So really shaping your end user license agreement to make sure that it encourages open sharing in the platform and more broadly with the world, while allowing and protecting the players from being, for example, exploited by industry partners potentially. But we think right now we're working really diligently to make sure that sort of everyone is protected and everyone has something to gain instead of leaving anyone too exposed. So the end user license agreement or something similar in different platforms, I think, are really key and something that we should definitely spend more time on, focusing on, you know, how do we protect the players and how do we protect the community to making sure that they feel protected and safe to, you know, contribute to this project, yet being able to control, you know, external factors and making sure partnerships are going smoothly and not held back by, you know, we want to keep everything internally and not share stuff. I think it's very key in citizen science to be as open as possible and to share your finding as openly as possible.
So great points. On the flip side, I want to ask Kara and Angela and Črtomir, how are you handling privacy? And maybe since Črtomir hasn't spoken yet, we'll let him start.
Okay, privacy is very important, but currently all our stuff is anonymous. So, but we need to work also with legal issues. In fact, now we are doing some virtual screening, but what if we will get some new innovative idea? So then the things are more complicated.
And
I do not know how we will solve this.
Okay. Well, I know in Europe, particularly with the new privacy legislation, the GDPR.
We have GDPR. We are not collecting any data about user. But maybe it would be wise to do this.
Yeah. So
we have no, in fact, we have no how to say statistics or something like this.
So with that, Kara, I noticed you're collecting phone numbers, and not in the US legislation, but in European, there also location data can be considered privacy, private data. So how, Angela and Kara, maybe Kara go first. How are you guys handling it?
Sure. So quickly on our website, we have terms of use and terms of service, as well as a privacy policy available for every user to explore and agree to before they submit data to us. And then in terms of research and sharing the data that we collect, particularly with public health and with our own researchers, we do anonymize all of that information so that it does protect the user in that sense.
Excellent. And Angela?
So what we do is we just remain anonymous. We never ask questions or ask for submitted data that would compromise that. All of the submissions are kind of fuzzed to a 10-kilometer, around a 10-kilometer space around where that user may have submitted. So we're hoping that in that way, people will feel safe enough to submit their data, and we're not holding anything, even like, the IP address from where somebody submitted in order to find out who that person is. So like, we can't backtrack to that person. I wanted to also just, speak to volunteers, because we are an almost entirely volunteer-run organization. We have maybe like, 5,000 people have built these BGIGs that I was telling you about. We have volunteers that drop in for a day. We have volunteers that maybe 100 hardcore volunteers that work day in and day out, and it's like their job. So we kind of just flow with that and try not to look at volunteering, intermittent volunteering, as a bad thing. It's like there's something there, there's an interaction there for anybody who wants it. So if you're just looking at the data and not contributing data, that's okay. If you're contributing data and you're really only interested in your data, that's okay too. If you want to help build new open data and open science mechanisms that would collect that data or help with the platform, we invite that. So there's a lot of different ways to contribute. But we definitely understand what the game players are experiencing in terms of like a flood of new interest, and then how do you maintain that interest, or how do you serve that interest, and what happens when it goes away. So those are just the two things that I wanted to remark on. But we are under the CC0. So under CC0, it doesn't matter how the data is used, but we will speak up if we feel like the data is being used inaccurately. So a lot of times, if people are cherry picking data and misrepresenting a situation, then we will, raise our hand and say, well, actually, if you look at the entire thing, or if you look at it within, with a greater scope, you will see that this is actually an inaccurate use of data. But other than that, if people want to use it for research, or if people want to use it to promote something that they can then sell, that's fine, as long as it follows that CC0 guideline.
That was actually one of my questions, Angela, is how do you handle making sure that your submissions are not, some jerk that decides to mess with your system, or there's some sort of that seems like that'd be a really hard problem.
Yeah, I mean, that could happen, right? But what we end up doing with radiation, the data submitted under radiation is, it's coming from, if you put the kit together and you submit the data properly, you could have outlier data, or you could have somebody mess with the data, but they'd be messing with the monitor as well. With air quality, we're really looking at that as a community experience. So, people could say, have their own air quality measuring device at their home, but what we're looking at is like community spaces where air quality would be measured, and then that they can see the air quality on a more granular neighborhood level, and see what their experiences are, but they're not necessarily doing that from their home. Some people are, but many people aren't. So in that way, you can say, like, how can data be your voice? How can data make you the subject rather than the object of research and science?
That's a fantastic ending point. Although I see we had one more question come in around data and data access that Dusoon was responding to online, so we can get it on camera for the record in the archive video. Dusoon, do you want to respond verbally to what you responded to Katerina in the chat box?
Sure. So to reiterate the question, it had to do with how the data generated by volunteers, which includes scientists as well as citizen scientists, expert researchers in the lab, as well as citizen scientists who are playing the game, how that's released and shared. And it's our goal at Eterna, and I think this is a goal of almost, if not all, academic laboratories, is to share experimental data openly and freely. So we strive to make data available, raw data, as well as analyzed data available through publications that are open access so that anyone can read them and access them. But also, parallel to the Eterna project, we've set up what we call the RNA mapping database, where not only us, but anyone who's carrying out these RNA structure determination experiments can deposit their data into a centralized location so you could access it. Any scholarly team may be able to access it. And we're still striving to share as much data as possible with the public.
Wonderful. So last question, if there's any questions from the audience, I don't know that I see any additional ones. You know, there's critics of citizen science, right? And particularly during this time, and questions about the statistics of what you're doing, etc. Would any of you like to respond to those critics and say, this is why citizen science is critical to our response efforts for COVID?
Yeah, sure.
Angela, and then Firas.
Sure. Thanks. Sorry, I didn't mean to jump in too fast. So what we found is that the more people participate, the more citizen science that there is, the more indisputable the data set becomes, because you have numbers on your side. So data then doesn't just become closed to university, government, or industry. Everybody is kind of cross-checking and generating that trust with each other. So I think, once again, if you have an open data set, you're not disputing what the data is, or if it's biased, you're just referencing like a common good. And when you create that common good, then that's when you say that it's not any individual that has to have a degree, but it's the quality of how people collect it and what they're and putting it out in the same manner that is important.
For us?
Yeah, I was just gonna say, you know, very generally, and I think it's quite evident in these times, these are really difficult problems that we're trying to solve. And I think just only relying on the traditional methods for solving them is not what we want to be doing right now. I think, you know, we want to be attacking these problems from every possible angle. And, you know, whether that's using citizen science, whether that's using another non-traditional method, I welcome it. Obviously, you know, it needs to be done properly, and there needs to be care when citing a lot of these things, as we see with, you know, news in general. But yeah, no, I would argue that, especially right now, we want to try everything. I mean, I think Bill Gates said it best recently, you know, in terms of trying to find a vaccine. They're going to build 10 different factories for 10 different vaccines, knowing that probably 9 of those are not going to work. But, you know, we need to find that one, and we don't want to lose any time just doing this one factory at a time. So I think I'll just leave it to that. I could go on forever.
Wonderful. Jasoon.
If I could chime in, first, I think not all the criticism is bad. I think critics really force us to identify weak points and know what we need to do to improve as a platform. So some of, you know, our most valuable improvements have come as a result of critiques. However, I think, especially in times like this, citizen science and citizen video games and these efforts generally benefit a lot from being an optimist, knowing that it may not be your actual effort that gets to the clinical trials, but knowing that you brought along all these people and increased accessibility to the in our battle against this pandemic, for example. I think almost everyone can agree that opening up accessibility to technology and STEM research is a good thing. And similarly with data sharing, I think allowing these more open platforms for data sharing and data recording are almost always a good thing if handled correctly, like Firas said. It's very important that you do not compromise on quality as you scale in quantity. But I think if done well, these efforts are going to really fundamentally reshape how science is done, how data tracking is done, how public health crises are handled. And I think this is a time where things are changing much, much faster than we can expect. And that pace of change is accelerating. And we need to be able to adjust as a society how to handle that pace of change and adjust to new normals. And I think citizen science will soon become a very commonplace thing in the scientific community and also the broader public health community as well.
Thank you so much. I think that wraps it up just beautifully. And we're now out of time, so I want to thank our wonderful panel today. I also especially want to thank Rhianna Putnam, who has done a tremendous job over this monthly series since the fall and helping us to organize this and run the meetings. And if you note in the side box, she's called out, please donate. It's not free to do this. The platform Zoom, for example, costs funding to do it, and as well as for Rhianna's time. So we really appreciate any donation you could make. And Rhianna has also included sign up for upcoming CSA online events. As she noted, there's several webinars coming up, including the one on May 28th on the role of citizen science for the Sustainable Development Goals. And with that, Rhianna, do you want to make any last comments?
No, I think you about covered it. You can register for any upcoming events on our website. And yeah, we will be sharing a link to the recording sometime next week, as well as all of the links that our presenters shared in the chat. And so if you have any follow-up questions or anything, we'll include some contact info so you can reach out to our presenters today.
And the last thing I would add is please join our Citizen Science Law and Policy Working Group. You can find the links. I think Rhianna posted them earlier, but we'll repost them and send it to everyone who's registered. Rhianna has also been kindly harvesting all the links that people put in the chat box, and that's going to be on a resource page combined with the links we've collected for projects during the prior webinars. And with that, thank you very much.
Great. Thank you.
Bye.
Thanks, everyone.